All right, good afternoon, everyone. Welcome to the University of Washington Department of Neurology Grand Rounds. There are a lot of new faces here today, and you guys are the future of the golden age of neurology. I'll say that since our chairman's not here. So welcome to the golden age. University of Washington is a great department of neurology, and we're really glad you're here, and hopefully you will like what we have here and consider us highly. Today, my job is to introduce our speaker. Our speaker today is Mike Crowder. He's the Alan J. Truer Endowed Professor and Chairman. He's the chair of the Department of Anesthesiology and Pain Medicine. He's an adjunct professor of genome science. He is actually the third Washington University person speaking. The first one was Dr. Ransom, who went there for M.D. Ph.D. Then the current chair of neurology, Dave Holzman, was here last week, so this is our third Washington University person. Dr. Crowder did his M.D. Ph.D. at Washington University, stayed there for his internship, came here for his residency, and then went back to Washington University. And that's kind of similar to what I did, bouncing between the two places. So there might be hope that someday I might be also chair. We'll see. But his talk today is Mitochondrial Proteostasis and Survival in Hypoxic Injury. So please welcome Dr. Crowder.
Well, I really appreciate the invitation to come talk about science instead of worrying all the time about budgets and non-clinical time and all the stuff that Bruce and I worry about. Seems like night and day. Again, thank you. This is just a pure science talk. I'm not going to talk about anesthesia care today, and I'm going to talk about recent work. None of it's published. It started in WashU, and I've brought most of my lab here, and we're still working on some of this. And some of this data is very hot off the lab. My lab is down at South Lake Union in what's called the Mitochondrial Metabolism Center down there. So first of all, I don't know if I have to do this, but I'll just say I have no conflicts of interest. And so hopefully this is an unbiased talk. And I want to start by talking about a pretty famous physiologist, Augustus Waller, who was doing experiments in his basement, apparently, as people did back then, and was transecting frog nerves, and noticed that there was this very characteristic degeneration that happened during sometime later after the nerve was cut. And he concluded, really with little evidence, but it turns out he was right, his instinct was right, that there was some nourishment coming from the proximal neuron that was going down the axon to keep the distal axon alive, and if you didn't have that, the distal axon died back. So this is where the term Wallerian degeneration comes from. It's observed in multiple diseases and conditions that you neurologists think about, I think, and see on a daily basis, like spinal cord injury, traumatic brain injury, peripheral neuropathies, chemotherapy-induced neuropathies, and even in Parkinson's disease and multiple sclerosis and Alzheimer's, there's evidence that there's Wallerian degeneration as a component of those diseases. So this is a very important problem, and a discovery happened about 25 years ago that really started to give us some insight into what was going on in Wallerian degeneration. It was a serendipitous discovery, as a lot of times the best discoveries are, and it was a group working in England and Oxford that was doing experiments in mice, cutting nerves and observing Wallerian degeneration. And they noticed that this strain of mice that they were working with, that the nerve wasn't dying back like it was supposed to, like they'd seen hundreds of times before. And if you look here, you can see that here's the normal. If you just look at action potential's ability to propagate down into the distal end of the axon, that after a couple of days, that's lost in the wild-type mouse. But in this unusual mouse, it remained for many days. So it was very distinct. It was really a dramatic phenotype that these investigators observed. And they later this locus where that was responsible for this, and it's segregated as a Mendelian single site locus. They named it the Wallerian Degeneration (WLDS) locus, and the game was on to try to identify what the WLDS gene was, or what the mutation was. So that it took about another decade for that to be discovered by the same group, actually, or a subset of that group. And it turned out to be an unusual mutation, a chimeric protein, where you can see that there's a fusion of something called, which was essentially ubiquitin type molecule, with a gene that is actually turned out to be Nmnat1, and there was a triplication of that, and it was basically due to a chromosomal translocation. So the question at that point was it the Nmnat gene or this ubiquitin gene that was causing it, or where it inserted, or something else? And so work was done in both fly and mouse, and ultimately showed that it was the Nmnat gene that was responsible. And so the kinds of experiments that were done were classic genetic experiments where you put in just the Nmnat, and you get, actually in flies, but not so much in mouse, you get a slow, a Wallerian degeneration defective fly. If you do something to the end of it, you get a Wallerian degeneration defective fly. But the bottom line of all this is that the Nmnat part is the active part, and the ubiquitin part wasn't essential for getting the phenotype. It did act like a gain of function. It was semi-dominant, and there was something extra that this ubiquitin part added to let the Nmnat be protective. So what is Nmnat? Well, it's an NAD biosynthetic enzyme. So that in this animal, you suspect that there's either overall more NAD, or that the NAD is higher in some compartment within the cell of these animals. And if you measure NAD levels, they're actually not higher overall, and they've not been measured to be higher in any one compartment. But it is clear that the biosynthetic part of Nmnat is required for this phenotype. So it's really thought that it's due to something, NAD being in a particular place within the cell that is protective from the Wallerian degeneration. So in the last decade and a half, the focus has gone more and more on mitochondria as the locus of Nmnat's protection. And so here's some of the data for that. First of all, if you get Nmnat1 is localized in the nucleus normally. If you get rid of that nuclear localization signal, so you get it out of the nucleus, it's more protective. Overexpression of a different isoform of Nmnat3, which is a mitochondrial localized Nmnat, is strongly protective. So it's suggestive that it's acting in the mitochondria. Axonomy produces mitochondrial loss and a decrease in mitochondrial dysfunction, and Nmnat actually ameliorates that. So it suggests that maybe it's doing this by making mitochondria function better or remain. And then if you reduce the mitochondria by a different method, this was done in flies, in the distal axons, the axons after they've been damaged or crushed, the protection by Nmnat is lost. So it says that the mitochondria need to be there for Nmnat to protect. So it's really pretty clear the mitochondria are the site of action of Nmnat in this phenotype, but it's definitely unknown how it works. Well, we became interested in Nmnat from work that was done actually in David Holtzman's lab, which I know David was here recently. And it was done in collaboration with Jeff Milbrandt, who's very interested in Nmnat and has been for a long time. And what they showed was that in a transgenic mouse that had this non-nuclear localized form, so getting Nmnat out of the nucleus. In that mouse that has less Wallerian degeneration, it's also hypoxia resistant. And this just shows, you can see that they call this cytoplasmic Nmnat, which is probably a misnomer, but that there's less stroke volume in the Nmnat, and you can see that here that the stroke volume is reduced in the Nmnat mice compared to the controls. So this is suggestive that this is not only protective against Wallerian degeneration, but it's protective against hypoxic injury. So Jeff Milbrandt and David Holtzman asked us if we wanted to get involved and study hypoxia, because that's what my lab does. And we mostly do that in C. elegans. But the first experiment we wanted to do was really convince ourselves that it was a hypoxic phenotype and not immune system or something to do with some other cell type, and not might be a glial phenotype. And we're not interested in glia. Sorry.
Lecture's over.
So we made pure neuronal cultures, mouse primary hippocampal cultures, and found that if you infected in with lentivirus, this non-nuclear localized Nmnat, you could get very good protection. So you can see that the survival of the cells is about double that in the Nmnat cells versus the control cells. So this was enough for us to say, okay, this seems to be a neuronal phenotype. It doesn't require glia, and worms don't have glia, and it doesn't require an immune system, and worms don't really have anything other than innate immunity. So we thought it was reasonable to try to study this in worms. So this is what we set out to do, a couple of years ago now, and here's the overall plan. We wanted to express the mouse non-nuclear Nmnat and C. elegans, and we were going to express it in two cell types, two types of situations, and all the cells in the worm are just in the neurons. And then we were going to ask overall whether the animal itself was resistant to death after hypoxia. And then we're going to look at other phenotypes just to see if there was anything else that we could study that might give us a clue to its function and how it was working. And then at that point test mechanistic hypotheses that we had formed. So we were successfully able to express the worm Nmnat the mouse Nmnat in a worm. This is the worm nervous system. There's 302 cells in C. elegans, and all of the cells seem to be expressing this Nmnat, which is has a fusion with m-cytNmnati, and then this is the ubiquitous form, and you can basically see it's in all cells. These are actually, this area right here is actually the germline. It's not expressed in the germline. So that was the first requirement, and then the next question was, did it have a phenotype? And we were very happy I wouldn't be talking about this if it didn't have a phenotype, and it had a very strong hypoxia-resistant phenotype in worms, just like it does in the mouse cells. And so this is the ubiquitous version. And N2 is the wild type worm. And this is Hypoxia survival. Taller is better. And these are basically two independent transgenic lines. This is actually just a single copy. Gives you very nice protection from hypoxic injury in C. elegans. What about the neuronal form? Same story, not quite as big an effect, but just protecting the neurons was good enough to protect the whole worm from death. So overall, the protection actually might be better in these cells, and we really don't know whether it's protecting non-neuronal cells or not. And then lastly, just for this kind of experiment, we took the worm version. We took there's two worm isoforms in C. elegans, and asked whether they're protective. So if you just overexpress the worm isoforms, and these worm forms do not have a nuclear localization signal, so they seem to be kind of ubiquitously in the cell. In both forms, this is one form here, and this is two lines from the other form. Both, if you overexpress in the worm, are protective. Now, we wondered whether the mouse version could actually act and replace the function of the worm in Nmnat. And that's important because if it can't do that, then you might argue that it's doing something that's really weird and it has nothing to do with Nmnat's normal activity. And we were able to ask this question because a null mutant in one of the versions of Nmnat in worms lays no eggs. It's completely sterile. And if you put the worm version back into that animal, you rescue the fertility as you would expect. And then if you put in the mouse version into that null animal, you get some fertility back. So this mouse gene is able to interact and do some of the normal functions of Nmnat as the worm version does. What about the other phenotypes? And why do we bother looking at the other phenotypes? Because we had what we wanted. We had hypoxic resistance, and why not just go for that? Is because these other phenotypes might be used as a surrogate to study the mechanism, and again, it might give us clues to the mechanism of Nmnat. So the most interesting phenotype that we found, and this was completely new, was that in Nmnat, the mouse Nmnat causes the worm to live longer. So it's got a long lifespan. You can see that in this strain, you see really quite a long lifespan versus the wild type form. But in this strain, you also see long lifespan as well. And the lifespan is nearly doubled in this version. So it's a big phenotype and very interesting for us. The other phenotype, which is really, I would say, more of a bother than anything else, is that overexpression of Nmnat actually causes some reduced fertility itself. So even though that mouse Nmnat could rescue the infertility of not having any Nmnat, too much is too much, and it actually causes problems in the worm. And I'll show you why. I'll show you why I'm bothering to tell you this a little later. Okay, so let's move on to the interesting stuff, the reason that we are studying this in worms, and that is maybe we can figure out a mechanism that hasn't been able to be figured out in fly or mouse yet. So what we did was just a candidate gene approach. That just means testing a lot of plausible mechanisms that could be responsible for one or all of the phenotypes that Nmnat mediates. And the way that we are going to do that is we've got the Nmnat hypoxia-resistant worm, and we're going to ask if a mutation in another gene affects that phenotype. So it either suppresses the phenotype or it enhances the And that would be at least a beginning of evidence that this gene functions in an Nmnat1 pathway. Okay, so we tested lots of genes, about 30 different pathways, and none of them had any effect on Nmnat's ability to make the worm hypoxia resistant except one. And the one pathway where we got 2 hits in the same pathway were what's called the mitochondrial unfolded protein response. So I didn't know what this was when my postdoc came to me and said, "Look at this." But he was smart enough to test this pathway, and it actually makes a lot of sense given what I told you before about mitochondria being probably the site of action. So what the mitochondrial unfolded protein response is just that. Mitochondria, just like in the cytoplasm, are really in all your cells. Your cells are constantly battling protein misfolding. There's a steady state level of misfolded proteins in all cells at all times. And the mitochondria is a compartment where that's no different. And when the level of unfolded proteins rises to some level that's, I presume, deleterious for the animal responds by trying to make chaperones and other sorts of proteins to restore what I call proteostasis, or improved sort of protein folding environment. This is a mechanism that's been studied a lot in C. elegans, so we're very well situated to study this in C. elegans. It's really, I would say, the premier organism to study the Mito-UPR. And so this is a pathway, a cartoon that's based on a lot of work in C. elegans. And I'll just start within the mitochondria, so you can imagine that, again, you've got misfolded proteins. The number of misfolded proteins rises, and there are proteases that start chopping up misfolded proteins. And some of these are called clip proteases. So that's shown by, I'm not sure why this is shown like a spool of thread, but for whatever reason, these proteases chop up the misfolded proteins. They're transported out of the mitochondria through at least one transporter that's known about, which is called HAF1, and I'll get back to HAF1 in a minute. So HAF1 is essential for this unfolded protein response, and essentially this black box from these peptides that are getting out of the mitochondria activate transcription factors. This is one type of transcription factor that I won't talk about today, but that does translocate to the nucleus and activates other co-transcription factors, and I will mention DVE1. And then they induce the transcription of these chaperones, HSP60 and HSP70, that then get transported back to the mitochondria and improve the folding environment. So that's the pathway. All right. So what my postdoc Mal found was that two genes in this pathway where there were mutations, where these genes had reduction of function mutations, actually loss of function. They were required when you made a double mutant with this Nmnat1 that makes the worms hypoxia resistant, and either HAF1 mutant or DVE1 mutant, so that you've got no HAF1 or no DVE1 activity, the level of hypoxia resistance went down a lot. And so here's, again, this is wild type, and these are some of the control strains, so they basically has no survival, particularly after hypoxia in these animals. Here's the ubiquitous mouse Nmnat1, gives you this very high level of survival from hypoxia. And then when you put it in the same background as this HAF1 mutant or this DVE1 mutant, you see a shifting back towards wild type, so genetic suppression. And the same is true, but to a lesser degree, for reasons we don't really understand, but it significantly suppressed the neuronal versions as well. Importantly, when we look at other resistant genes that we've discovered over the years where there's mutations that make the worm resistant, it doesn't suppress those. So it's not that it just suppresses everything that makes a worm resistant to hypoxia. It's at least specific to this degree for Nmnat. And this is why I bothered to tell you about the infertility, is that it also suppresses the infertility phenotype. So here you can see that there's reduced number of eggs in this ubiquitous Nmnat compared to the wild type, and that there's a significant improvement in fertility going from here to here when you have that half-1 mutation there. So we were wondering, well, maybe this is an effect that's indirect due to the fertility and not due to something more interesting. But we know that's not true because we have strains that are normally fertile where half-1 suppresses their hypoxia resistance too. So anyway, there's something fundamental about what Nmnat is doing that the half-1 mutants, half-1 and DV1 mutants, the mitochondrial UPR mutants, suppress. Likewise for lifespan, and even though the effect is not as dramatic, it is highly significant. So here's the wild type, here's the Nmnat animal, and here's the Nmnat animal with the DV1 mutation. So significant suppression of the prolonged lifespan phenotype. Okay, so this work basically up to this point we've just recently submitted for publication, and what's next is what we're working on now, which is basically what this told us, and that is a broader question or broader hypothesis, and that is that hypoxia actually reduces mitochondrial protein misfolding. And that's not something that anyone has published or talked about as a way that hypoxia might be producing its phenotype. And that activation of the unfolded protein response, which improves the folding environment, should be protected if this is true. And then back to Nmnat, that Nmnat provides hypoxia protection by actually activating the mitochondrial UPR. So that's our hypothesis. This is kind of a three-pronged hypothesis. And there's predictions from the third part of the hypothesis, and that is that the Nmnat worms should be resistant to other ways other than hypoxia that produce mitochondrial folding stress, and that Nmnat should actually induce the mitochondrial UPR. Okay, so that's the rest of the talk is going to show you data for each part of this, hypotheses. So first of all, hypoxia does most definitely induce mitochondrial protein misfolding, and we looked at this in 3 different ways. So first of all, there's a dye that's a hydrophobic dye that binds to membranes, but inside the cell it doesn't bind to much because there's not that many hydrophobic surfaces inside your cell or inside your mitochondria. And and ANS is actually relatively selective for mitochondria, and you can see that after hypoxia, and by the way, this is just a very mild hypoxic injury. These animals are actually behaving almost normally. None of them are dead, and yet they have this profound increase in the amount of hydrophobic surfaces in the mitochondria after hypoxia. The second way that we looked at this is we literally just fractionated, we basically purified mitochondria and asked if there's an increase in tryptophan-soluble protein in animals that have seen hypoxic injury and worms that have seen hypoxic injury. And there's about a 40% increase in the amount of tryptophan-soluble protein after hypoxia in the mitochondria. And then the third indicator, which is admittedly an indirect one, is that hypoxia actually does induce the mitochondrial UPR, the sort of transcriptional output of the mitochondrial UPR, which is these chaperone genes. And so we used a GFP reporter attached to the promoter of this gene, and so it measures the transcriptional output of this gene, and you can see that even though the data is fairly messy, that there's a significant, highly significant effect on induction of this mitochondrial chaperone that's responding to misfolded proteins in the mitochondria. And then the last way, which I just like the most, is electron microscopy, where we see this really amazing aggregates inside the mitochondria. And again, these animals are relatively healthy. They're not dead animals. They're not very sick from the hypoxia, but they've seen enough hypoxia to cause some issues. And you see these aggregates inside the mitochondria here, and this is a quantitation of that. You can see that about half the animals have some aggregates in their mitochondria after this mild hypoxic injury. So definitely hypoxia induces a misfolding, bad folding environment inside the mitochondria. Okay, so the next part of the syllogism is that mitochondrial UPR induction protects against hypoxic injury. And the way that we've done that is because there's been a lot of work on mitochondrial UPR and C. elegans, it's known how to induce it independently, not with hypoxia. And so one way you can do this is essentially to reduce one of the subunits in the electron transport chain so that there's a stoichiometric mismatch between this CCO1 subunit and the other subunits in the electron transport chain. So this causes misfolding because there's not enough partners now, presumably, to for them all to be folded. And that induces the mitochondrial UPR. And you can see that when we do RNAi with CCO1, so we're reducing its levels in the mitochondria, we get a big effect on survival. So there's dramatic improvement in survival. And this effect is suppressed by a transcription factor in that mitochondrial circuit, the mitochondrial unfolded protein response circuit. So we know that the induction requires, the protection requires the mitochondrial UPR. Now, you may be asking yourself, well, you're knocking down the electron transport chain, and so you're affecting ATP levels, you're reducing oxygen consumption, and so maybe why these worms are hypoxia resistant is they're just better able to hold their breath, if you will. They don't, they're not using ATP as rapidly. There's a couple of ways that we know that that's not the explanation. One is that we can knock down other things that have nothing to do with the electron transport chain, and they also produce resistance to hypoxia. And two of those have no effect on oxygen consumption. Now, CCO1 does. If you knock down an electron transport chain protein, you do get reduced oxygen consumption, but with those you do not. All right, so the next step in looking, testing our hypothesis is to ask whether activation of the mitochondrial UPR after injury also improves recovery, and this is something that we've worked very hard to be able to do in worms. Is be able to do what you really want to do. If you're a stroke neurologist, it's very rare for you to know a stroke's going to happen, so that you can give them their TPA or whatever before the stroke. You need to give them a drug after the stroke for it to be effective. And this data shows that, in fact, activating the mitochondrial UPR with doxycycline or with CCO1 RNAi after the hypoxic injury improves survival, improves recovery from hypoxia. So we needed a different way to induce mitochondrial folding stress. One way that's been used routinely in C. elegans is ethidium bromide. That's stuff that if you've done DNA gels, you've probably used ethidium bromide. So ethidium bromide binds to DNA, but it binds to mitochondrial DNA better than it binds to nuclear DNA, and it will cause a mismatch between nuclear encoded subunits in mitochondria and mitochondria encoded subunits, and has a dramatic induction of the mitochondrial UPR and a dramatic misfolding effect. And so if you grow worms on a plate that has some ethidium bromide in it, they basically arrest. They don't grow up. They don't reach adults, and so that's what's shown here in a wild-type animal. You get very few adults on an ethidium bromide plate, but you can see both ubiquitous and the neuronal forms of Nmnat expression allow the animal to grow up, and so that suggests that Nmnat is protective against mitochondrial folding stress just in general. And then lastly, Nmnat1 modulates the mitochondrial UPR, and that's obviously has to be the case for this whole working hypothesis to be reasonable. And so what this is this essentially a reporter strain that has, again, it's that transcriptional reporter for one of those chaperones that's in the mitochondria. So this is a measure of transcriptional activity at that gene. And so this is sort of basal activity, and this is the activity in the Nmnat1 strain. So it's a huge induction of that for both this reporter and a distinct reporter. This is yet a different mitochondrial chaperone. And if this all makes sense, then it should be the case that the mitochondrial UPR gene that is required for the hypoxia resistance of Nmnat is also required for the UPR induction by Nmnat, and that's what this data shows. So here's induction by Nmnat, and here's suppression of induction by the DV1 gene mutant. So this is the data I wanted to show you today, and I just want to summarize by saying that Nmnat is seemingly a cure-all molecule. It reduces axonal degeneration in mouse, fly, and I didn't show you the data, but we have recent data that Taxol-induced axonal degeneration in worms is prevented in these Nmnat animals in worm. It improves survival from hypoxia, and we know that in mouse and worm, and it prolongs lifespan, and that's something that's only our data at this time. So there's several lines of evidence that I gave you from other people's work that says that the mitochondria is probably the site of action of Nmnat, and that's what makes it reasonable what we've discovered, which is that Nmnat enhances response to mitochondrial folding stress and enhances this mitochondrial unfolded protein response. And this action is essential. Our genetic data shows that this pathway is required for the protection. And then a completely novel concept is that hypoxia induces mitochondrial protein misfolding. It's really not surprising. It's just that people haven't observed it. And that mitochondrial unfolded protein response is hypoxia protective. So doing something to mitigate or deal with misfolding in the mitochondria is protective against hypoxia. And so the mitochondrial proteostasis is a reasonable therapeutic target to look at to see if we can modulate that, we might find a distinct way to protect from hypoxic injury. So all of this work was done by two people. So the very talented faculty member now, he's the one member of my lab that didn't come with me to UW. He had ties to St. Louis and just couldn't leave. But Mal is now working with Jeff Milbrandt, so the people that the lab that convinced us to get into this NmNAT work in the first place. So he's back in St. Louis in the genetics department. Daniel Kaufman is an MSTP student, a WashU MSTP student that's in my lab here. He'll complete his thesis here and then go back to WashU to do his clinical rotations and graduate in a couple of years. And Daniel did all the sort of non-NmNAT work that I talked about, the sort of more fundamental work that I talked about. I want to thank Jeff Milbrandt and his lab for convincing us to do this work and giving us advice and reagents, and our funders, currently NIH and former funders, especially the McKnight Foundation and IRS, which is the International Anesthesia Research Society. So thank you very much. And I will take questions, and apparently people can type in questions. Yes, yeah.
Changing the mitochondrial UPR affects the mitochondrial.
So the question is, I'm told to repeat the question. So the question is, does the mitochondrial lymphotic protein response affect mitochondrial membrane potential? And I'm sure that if you have a dying mitochondria, it will. But with mild proteostatic stress, we see no effect when we look at membrane potential sensitive dyes in the mitochondria. We see no effect. Yeah.
Is there an interaction with the release of cytochrome C?
Yeah, we don't know. We don't know what this does to cytochrome C release. Cytochrome C release is not required for apoptosis in C. elegans. It's not the mechanism of apoptosis in C. elegans, even though the pathway for apoptosis was identified in C. elegans. It's weird. It's not like mammalian apoptosis, and so there's not, other than actually measuring it in some way, we don't have a sort of a genetic way to look at that, but so we don't know. Yeah.
I guess we've been trying to pin you down about what's the downstream impact of initiating UPR interfering with it. So how do you?
Yeah. How is it protective?
How is it protective?
Yeah. I think we don't know. We presume that it allows the mitochondria to survive. If you look in C. elegans in our model at the mitochondria after hypoxia, most of them are gone. There's very few mitochondria that stain. We don't really see them with a severe hypoxic injury that actually kills the animal. But we don't know if it's more specific than that. It could be that there's some very specific pathway that we don't know about or that anyone else knows about that causes the death of the cell. If it's cytochrome C or something bursting from the mitochondria, we don't know.
I was curious as to whether the original Oxford mice were sort of super mice in some way.
Do they have a long lifespan?
They are, or
Yeah.
are they resistant to various things?
Yeah, so the original mouse, the Wlds mouse, has not been tested for hypoxic resistance or life, and no one knows about the lifespan. I mean, the few people that I've talked to know I guess the reviewers that are reviewing our paper now know, but that's about it. So we haven't tried to do lifespan experiments in mouse. We can't. That's not our gig, and we can't afford it, basically. So that's, you know. Uh-huh.
Is mitochondrial UPR more sensitive than the whole cell UPR?
Yeah. So the question is, the mitochondrial UPR more sensitive to hypoxia than other unfolded protein responses in the cell? So we also know that the ER, the endoplasmic reticulum unfolded protein response, is sensitive to hypoxia. It's induced by hypoxia, and we've actually got several papers on that, and we initially tested all of the genes in the ER unfolded protein response to see if they suppressed, because they do suppress other. They are required for other hypoxia-resistant genes that we've looked at, but none of them suppressed. None of them had any effect. In terms of actual sensitivity, they're basically induced sort of similarly. So kind of at a similar dose, if you will, of hypoxia. So I'm not sure that one's more sensitive than the other, but this phenotype is much stronger than when we try to upregulate the ER UPR. We actually don't get a very strong protective effect from upregulating. It's needed for hypoxia resistance of some of the pathways that we found, but upregulation doesn't do much for that.
Is there much known about the comparable sites in the human genome?
So as much known about the Mito-UPR in the human genome. So both mouse and human have 3 Nmnats, 1, 2, and 3, cleverly. And you've heard about 1. 1 is primarily nuclear localized. Nmnat2 is primarily in the Golgi and the ER. Not clear what it's doing there. And the third one, Nmnat3, in mouse at least, is in mitochondria. I actually don't know if it's known. I presume it is known. I just don't know whether Nmnat in human cells is also in the mitochondria. Has anyone seen a missense mutation or something in Nmnat or something about Nmnat that gives them an interesting phenotype or makes them long-lived? No. Yeah.
Nobody's looked at polymorphism.
I'm sure people look at polymorphisms in every site, but I do not know. There's no published papers that I'm aware of that have linked in any of the loci, in any of the Nmnat genes to an interesting phenotype in humans.
So you stated that mitochondrial proteostasis is a reasonable goal. Could you comment on kind of the thoughts how you would want to achieve it, or?
Yeah. So the things that if you
repeat the question.
thank you for reminding me. Yeah. So the question is, if I believe that mitochondrial unfolded protein response is promising as sort of a lead idea for therapy, how would you do that? Well, one of the good things is that we know if you activate it after hypoxia, it works, it's protected. So that's kind of essential. That's part of things that we do with everything that we discover that's protected from hypoxia in worms is to ask if it works afterwards. The next step is to find something that's actually a lead compound that activates the mitochondrial UPR. And well, we've got doxycycline, we've got ethidium bromide. So I don't think we're going to be giving ethidium bromide to patients. Doxycycline maybe, but people are already looking at, people have already looked at doxycycline for stroke. And so I don't see that as, we won't do that. And so what we plan to do, and we're actually doing this now, is we're doing small molecule screens with the help of the screening core at South Lake Union which can do some amazing things. So they basically, in a 384 well plate, can give you about 10 worms that have seen hypoxia, and we have a way for them to slowly die over the next 5 days, and then they put a drug in there and ask if it survives. So we're doing that, but that's kind of a general screen. It doesn't necessarily have anything to do with mitochondrial UPR. But the next screen that we're gearing up to do is use that reporter, that GFP reporter that is transcriptionally upregulated, and so you get a green worm if you have induction of the mitochondrial UPR. And so we're going to look for molecules that induce the mitochondrial UPR with this. And that'll be a very easy screen because it'll be entirely in silico from our point of view. We'll basically hand them a big tube of worms, and then a day later we'll have the answer.
Kind of a follow-up question to that, how long would you, what do you envision? Would you give the drug for a week, 10 days?
Oh, well, I have no idea. Yeah, I mean, if we get to that point, I'll be a happy man. Yeah. Okay, thanks a lot.